As people, our needs consist of food, shelter, fuel, health, and a variety of material objects, and most of these are controlled by what our environment can provide, as well as how our body can adapt and use such environmental resources. But as populations increase and society becomes more complex, our resources have seemed to become more limited in what they can supply. Until now, humanity has been trying to get by using expensive and ineffective methods to meet our needs, but not anymore. Hi, this is Rosie from Genetech 75, and today we're asking for a $1 million investment in our product, bacterial transformation, a technology that will disrupt how society will operate. Essentially, we're harnessing the power of bacteria to create the biological molecules that we need by feeding them the instructions. These instructions come in the form of plasmids, DNA fragments carrying genes that can replicate independently. But the best part is that we can also piece together our own instructions through recombinant DNA, in which DNA from different sources artificially replace old DNA. In the past, competitors have been able to construct their own plasmids. However, our company will be the first to create a formula to introduce these plasmids into bacterial cells. In our experiment, we used the Pico plasmid, which consisted of a DNA sequence called GFP, responsible for producing a green fluorescent protein, as well as BLA, responsible for producing an enzyme for antibiotic resistance called beta-lactamase. Now, to simplify the explanation of our innovation, I'm going to use an analogy. As I am your average teen, I'll be using Nintendo's Kirby. Kirby is a character who gains their powers by absorbing powerful objects in their environment. The higher Kirby's level, ranging from 1 to 5, the better Kirby's ability to inhale surrounding objects. In this sense, our E. coli bacterial cells are naturally much like a level 1 Kirby, and our Pico plasmid is a powerful object that carries two new traits. First, the ability to make Kirby glow fluorescent green thanks to the GFP recombinant DNA on this plasmid, and two, the ability for Kirby to be completely resistant to an enemy, ampicillin, thanks to the BLA DNA on the same plasmid. In this procedure, we force a level 1 Kirby to become a level 5 Kirby, and then ensure that our Kirby recovered to survive. We used two microcentrifuge tubes in this experiment, labeled plus DNA and minus DNA, in correspondence to whether or not the Pico plasmid would be added. First, 250 microliters of calcium chloride was added into the two microcentrifuge tubes using an F250 micropipette. Then we transferred 4 large colonies of E. coli bacteria, or Kirby's in our case, to each tube from a source place using an inoculating tube, twirling them in the solutions to dislodge all the cells. Next, 20 microliters of Pico plasmids were added into the microcentrifuge tube labeled plus DNA using an F20 micropipette. Now, this step was important because the phosphate groups in the plasmid DNA and the phospholipids of the bacterial membranes are naturally negatively charged, and so they would repel each other. Calcium chloride is added because when the compounds dissociate, the positively charged calcium ion neutralizes these electrostatic charges, increasing the chances that the plasmids and the bacteria come closer in proximity. This is important because our bacteria, just like Kirby, cannot engulf objects that are too far away. The presence of calcium chloride thus upgraded our Kirbys to a level 2, since it increased the likelihood for them to take in the plasmids. Next, the solutions were incubated on an ice rack for 10 minutes. The cold made all these activities in the solution slow down significantly. As a result, this also freezes open the naturally occurring pores in the bacterial membrane, which increases the likelihood for the plasmids to enter the bacteria. Immediately after, we then heat shock the solution by incubating the solutions at 42 degrees Celsius for exactly 90 seconds. As heat is added, the solution outside of the cell heats up. But given the short time frame, the inside of the bacteria cells still remain cold, creating a temperature difference. Since fluids move from hotter to colder areas, the contents outside of the bacteria, such as the plasmids, move into the cell. We found that 90 seconds created the best temperature difference to create this convection current. We used 42 degrees because it's hotter than the temperature that our E. coli are used to, but not too hot to a point in which they'd heat up too much. This step forces our Kirby's to a level 5 since they're essentially force-fed as many plasmids as possible. Finally, in this transformation process, the solutions were incubated on the same ice rack again for another 2 minutes so that they would cool down, preventing the newly introduced plasmids from leaving the bacterial cells. Now, the bacterial cells were just put under a lot of stress, and this makes them really unhappy, but we need them to stay alive and return to their normal function so that they can produce proteins. Now, the E. coli bacterial cells were just put under a lot of stress, which probably made them really unhappy, but we need them to stay alive and return to their normal function in order to make proteins and reproduce. To encourage this, 250 microliters of a lysogeny broth was pipetted using an F250 micropipette into both tubes, which were then incubated at 37 degrees for 20 minutes. The lysogeny or Luria recovery broth consisted of yeast, amino acids, nucleotides, salt, and vitamins, which increase the abundance of key ingredients necessary for bacterial reproduction and protein synthesis. In addition, 37 degrees is the optimal temperature for the bacterial cell's activities. This step was especially important because we wanted to ensure that the bacteria who took in the plasmids had time to use the plasmids and produce beta-lactamase, the protein responsible for ampicillin resistance. This is because next, 250 microliters of each of the final solutions were dispensed and streaked onto agar plates that contain ampicillin. Without the last step, the bacteria would have died immediately since ampicillin is an antibiotic. Ampicillin was on the plates to ensure that only the bacteria that had taken in the picoplasmid and used it to make proteins could survive and reproduce. These would be the bacteria who acquired antibiotic resistance from beta-lactamase production, as well as the ability to glow green. We've named this technique selective plating, as our ultimate goal was to assure that the resources were saved for only the desired bacteria. Finally, the plated bacteria were incubated at room temperature for 24 hours to allow for replication. When we first presented our procedure, our competitors didn't think that we'd be successful. However, we were confident in our method. We predicted that if we were to plate the transformed E. coli bacteria onto an ampicillin plate, we'd only find bacterial growth that would glow green under fluorescent light, since these bacteria would have acquired this trait along with the ampicillin-resistant trait, just like how Kirby gains abilities from the abilities of objects that it absorbs. Similarly, we predicted that the plate with untransformed E. coli bacteria would produce no growth since they would die from the lack of ampicillin antibiotic-resistant properties. Much to everyone's surprise, our results matched our prediction. Of course, we tested our method multiple times and our results were even better than expected. Not only were our predictions correct, but our transformation was highly efficient, too. How do we know this? Well, we used a simple formula to calculate it. Transformation efficiency, measured in transformants per microgram, was calculated by dividing the total number of transformants by the mass of the P-glow used, times the total volume of the solution divided by the volume plated. We counted around 1,200 transformants on our agar plates, and the mass of the P-glow was calculated by multiplying its concentration by the volume, which produced 1.25 micrograms. The solution volume was the sum of the volume of calcium chloride plus the volume of piglo plus the volume of lysogenate broth used, which was in total 520 microliters. Finally, the volume plated from our procedure was 250 microliters. In total, our transformation efficiency was approximately 1,996.8 transformants per microgram. And this was really impressive, if you ask me. Now, we've proven to you today that our product is legitimate. You might still be wondering, however, why are we worth a $1 million investment? My answer is simple. The power of this technology stretches far beyond just changing the appearance of E. coli, as it has many applications as well. For instance, the hepatitis B vaccine is made by helping individuals build an immunity to a protein found on the virus's surface, providing protection against infection with the hepatitis B virus. Our first hepatitis B vaccines were made by taking blood from samples from people infected with the hepatitis B virus or purifying the surface protein from the infectious virus. However, this process was strenuous, expensive, and people risked contamination due to the use of blood. Using our technologies today, we can take the part of the virus that makes the surface protein gene and put it into yeast cells. The yeast cells can then produce many copies of the protein that are subsequently used to make the vaccine, using the same mechanisms that we did in this procedure. By manufacturing the surface proteins in laboratories, we could ultimately reduce the costs and increase the efficiency of the hepatitis B vaccine. And the benefits to bacterial transformation don't stop there either, as it can greatly impact a number of major industries as well, such as agriculture, bioremediation, and medicine. And with your investment, we could expand even further. Once again, bacterial transformation is our product from Genetech 75. Thank you for considering our pitch.